Hello everyone, I am one of the biology faculty member at Education World Gurdaspur, one of the famous and renowned institute in the Gurdaspur city in Punjab. And today my presentation basically deals with the study of various kinds. In the previous lecture which has been uploaded on Education World YouTube channel with the title name Education World EW, I have discussed about the blood composition, and today I will discuss about the various kinds of formed elements and blood cells in the human beings. So now if you see on the first slide, you may see diversity in the blood cells, the varied kind of the blood cells you may visualize on the screen. So keep in mind RBC or erythrocytes, these are the smallest kind of the blood cells, and these are much abundant of other categories of blood cells as well. And WBC, they are larger in size in comparison to the RBC, and they may show diversity in their form, size and shape. For example, the lymphocytes are the smallest kind of the, and monocytes are the largest kind of the. And the third type of formed elements which are reported in the blood plasma, these are known as thrombocytes or platelets. These are not the true cells, rather than these are the cellular fragments. These are the fragments of megakaryocytes. These are the large cells of the bone marrow. The megakaryocytes, they split, undergo fragmentation and forms the platelets. So now, if we first of all move to the first category of blood cell, that is the RBC or erythrocytes. So with regard to RBC, I have already discussed with you, these are the most abundant kind of the blood cells. That's why our blood is having the red color. And it is biconcave in shape. That is the central part of the RBC is thin in comparison to the peripheral part. In other words, I can say the periphery of the RBC is quite thick in comparison to the central area. So this is called as a biconcave shape. And this shape is so flexible that the RBC, they can pass through the smallest blood vessels, that is the capillaries. The average diameter of the RBC is 7.5 micrometers, and they are 2 micrometer thick at the rims. And we have already discussed these are smaller than the WBC. The size of the WBC ranges from 12 to 20 micrometer in comparison to the RBC, who are having the size range of 7.5 micrometer. So RBC, they are biologically significant type of cells, as they contain a wonderful conjugated protein known as hemoglobin. The hemoglobin is actually a conjugated protein which is formed by the fusion of protein and non-protein moieties. The non-protein part of the hemoglobin is known as heme, which is actually iron 4-ferrin complex, and globin is the proteinaceous portion of the hemoglobin. This globin portion is comprised of 4 polypeptide chains, 2 alpha and 2 beta. Each alpha chain is having 141 amino acids, whereas each beta chain is having 146 amino acids. On the whole, a globin molecule in the hemoglobin, which is made up of 4 polypeptide chains, is having total 146 into 2 plus 141 into 2 equivalent to 574 amino acids. That is the globin portion of the RBC contains 574 amino acids. And in chemistry, the hemoglobin molecule is regarded as a coordination compound. It is a coordination compound of iron. And the biological function of the hemoglobin is it has affinity for binding with the oxygen, and in addition to oxygen, it can bind well with the carbon dioxide and carbon monoxide. So primarily, in the normal conditions, it helps in the transportation of oxygen by forming a complex with it. The complex of oxygen and hemoglobin is known as oxyhemoglobin. Then, the young RBC are nucleated. They have all cell organelles, but the mature RBC lost its nucleus as well as the organelles as to accommodate more amount of hemoglobin. Mature RBC is enucleated. So I have already discussed the structure of the hemoglobin with you. It is having protein as well as a non-protein portion. The non-protein portion is known as a heme, and proteinaceous part is called as a globin. And this globin is made up of 2 alpha chains and 2 beta chains. In other words, the symbolic presentation for the globin part of the hemoglobin of adults is alpha 2 beta 2. That is, it is having 2 alpha chains and 2 beta chains. So keep in mind, the fetal hemoglobin is somewhat different from the adult hemoglobin, because the globin part of fetal hemoglobin is having the symbolic presentation as alpha 2 gamma 2. That is, again, it is made up of 4 polypeptide chains. The 2 chains are alpha, but other 2 chains, they are gamma type, they are not of beta type. And fetal hemoglobin, which is represented by the formula HbF, it is having comparatively many-fold more affinity than the adult hemoglobin. So you may see here the various components, and this is the non-proteinaceous portion of hemoglobin, which is called as a heme. And each heme group bears an atom of iron, which binds reversibly with 1 molecule of oxygen. Then, one of the important chemical property of the hemoglobin is hemoglobin has extremely high affinity for carbon monoxide, moderate affinity for carbon dioxide, and less affinity for oxygen molecule. I want to say, if carbon dioxide is there in the air, then hemoglobin within our RBC tries to bind with the carbon monoxide in comparison to oxygen. So this may lead to the carbon monoxide poisoning, and the person may suffer from the suffocation and painful breathing. So this may lead to the death as well. So we can say the carbon monoxide deoxygenate hemoglobin by binding with the hemoglobin, and its treatment is the patient must be supplied with the oxygen in the hyperbaric oxygen chamber. Then when the hemoglobin binds with the oxygen, it will form a complex which is called as oxyhemoglobin, and it becomes red in color. But when it gets deoxygenated, then it becomes dark red in color. And carbaminohemoglobin, which is a complex of carbon dioxide and hemoglobin, that is also formed during the respiratory transportation of the gases within our blood. So now, if we discuss the functions of RBC now, the main function I have discussed in their biology. The main function of the RBC is to transport oxygen from lungs to the tissue cells and carbon dioxide from tissue cells to the lungs. Secondly, the RBC contains hemoglobin, which acts as excellent blood buffer. That is, it retards the alteration in the pH of the blood. So with regard to the formation of RBC, from where the RBC are formed, so the process of formation of the RBC, first of all, it is known as erythropoiesis. It is a part of hemopiosis or hematopiosis, which refers to the whole blood cell formation. So for your kind information, the RBC, they are formed in the fetus from yolk sac, which is one of the embryonic membrane. And after the birth, the RBC or blood cells, they are produced by the liver. And in the postnatal life, in the childhood, when the bone marrow gets developed at an age of 1.5 years, then the process of formation of RBC and other blood cells is grabbed by the bone marrow. I want to say, in the adults, the bone marrow is responsible for the formation of the various kinds of blood cells like RBC, WBC and platelets. So within the bone marrow, which is a fatty vascular tissue inside the long bones such as a femur and humerus or in the vertebrae, the fatty vascular tissue is there, and that fatty vascular tissue is known as a bone marrow. The bone marrow is having the stem cells. Stem cells are actively dividing cells. They divide and produce the new blood cells. And bone marrow is of two different types. One is called as a red bone marrow, second is called as yellow bone marrow or white bone marrow. The red bone marrow is located in the shaft part of the bone, whereas The yellow bone marrow or white bone marrow is restricted in the apophysis or ends of the long bones. And on average 1 ounce or 100 billion blood cells they are formed each day from the bone marrow. This is called as a physiological regeneration. So this red bone marrow is named so because it is having much more amount of vascular tissue in comparison to fatty tissue. So you may see this is a flow chart, diagrammatic flow chart showing the formation of the various blood cells from the bone marrow cells, stem cells. The stem cells in the bone marrow, they are known as the hemocytoblast. The hemocytoblast, they divide to produce the various other kinds of daughter cells which make as precursor cells for the WBC, for the platelets, for the RBC, and the RBC precursor cell is known as pro-erythroblast, which undergo a sequential change and finally matures into the erythrocytes. You may see the various steps in the process of formation of RBC. First of all, the hemocytoblast, which is located in the bone marrow, it converts into the erythroblast, then into normoblast, then into reticulocyte, and finally into the erythrocyte. And RBC, they are produced throughout whole life to replace the dead cells. The lifespan of the RBC is 120 days, and in the meantime the new RBC, they are formed by the bone marrow. So there are certain control mechanism also which can regulate the process or rate of formation of RBC. That is, there is some factor that is responsible for controlling the rate of erythropoiesis. So primarily during the oxygen suffocation conditions, what will happen? The kidneys secrete erythropoietin hormone. This erythropoietin hormone stimulates the bone marrow to produce more amount of RBC. So this happens when the oxygen scarcity is there. That is, I want to say, when you move to some hilly area where the oxygen pressure is less, then the kidney gets stimulated to produce the erythropoietin, and erythropoietin then stimulate the bone marrow to produce the RBC. And the drop in the renal blood oxygen level can result from hemorrhage or excess RBC destruction or reduced availability of oxygen to the blood, as might occur at the high altitudes or during pneumonia, and increased demand for oxygen commonly in those who are engaged in aerobic exercises. So what are the various ways to increase the RBC count? First of all, in the sports person, and legal method is by training athletes at the high altitudes, and illegal is use erythropoietin, androgen or other hormone analogs as injections in order to stimulate the bone marrow to produce more number of RBC. So the dietary requirement for the erythropoiesis are iron, vitamin B12 and folic acid, and more important to the woman due to the loss of blood during menstruation. So then the average lifespan of the erythrocyte is 120 days, and when the RBC, old RBC, they are degraded in the spleen, then what will happen? The raw material again get regenerated, that is a hemoglobin. They are further broken down in order to get the new material for the formation of new RBC. And there are a few erythrocyte disorders such as anemia, a condition in which the blood has abnormally low oxygen carrying capacity due to the lesser number of RBC. And common causes of anemia include an insufficient number of RBC, decreased hemoglobin content, abnormal hemoglobin, and I can cite the cases of abnormal hemoglobin by the examples such as thalassemia and sickle cell anemia. Another disorder of RBC is Polycythemia, and this is the disorder in which the number of the RBC get increased, and it increases the viscosity of the blood, causing to sludge or flow sluggishly. And common causes of the polycythemia includes the bone marrow cancer and a response to the reduced availability of oxygen at the high altitudes. So today I will discuss with regard to the RBC. In the next lecture I will discuss about the human blood groups, about WBC and other kinds of blood phenomena. Thank you so much. And if you like this presentation, if you like this video, kindly subscribe our channel Education World EW on YouTube, and share this video, like this video, and I'm hoping for your positive response. Thank you so much.